So hello, good morning, everyone. And we fully realize it's early morning, but we need to get started. Today, I really thank everybody for coming back. We had a fantastic scientific day yesterday. I think it was absolutely wonderful to speak, learn so much about science. And I really thank everyone involved yesterday in all the talk, chairing, and all the remaining event. I would like just to remind everyone, particularly the chairs, to really track the time, and also the speakers to be on time. We have a very intense program, and it's very important to give to everyone their chance to and their time to speak. So please be on time. We really appreciate this. And with this, I would like to transfer the mic to Musa, who will introduce the second plenary speaker.
Thank you, Piero, and very good morning for the second day of the Human Cell Atlas General Meeting. And it gives me a huge pleasure to introduce Professor Yasmine Belkaid, who is unfortunately unable to be with us in person today and will be joining us despite being ill from Paris. And so thanks very much, Yasmine, for making it this morning and to give us your talk. Just a bit of background about Yasmine, who was born in Algiers, in Algeria, and graduated with from the University of Science and Technology, Hori Boumediene in Algiers, as well as a master's from Paris Sud University, where she did her PhD there at the Institut Pasteur on Leishmania. She then moved on to the US, where she stayed at the NIH, specifically in NIAID, and since 2009 was leading the Laboratory of Host Immunity and Microbiome. Yasmine is currently the president at the Pasteur, and her research and her achievements have been hugely acknowledged in the forms of many, many prizes, including the Robert Kock, Lurie, and the Sanofi Institute Pasteur Prize, and she is on the has been elected to the American Academy of Arts and Sciences, the National Academy of Medicine, and the National Academy of Sciences. Yasmine also was on the NIH Anti-Racism Steering Committee, and she's also on the Genentech Scientific Resource Board. And it's a huge pleasure again to have Yasmine join us today on Zoom, and we look forward to your talk today. Thanks, Yasmine.
So the first thing that he did was really to first look at the evolution of the mammary gland, and in particular the mammary epithelial cells, or these new epithelial cells that are really required for the contraction of and the release of the milk, and looked at the accumulation of lymphocytes within the tissue. And what he was able to show is that it was a parallel between the accumulation of those cells and the lymphocytes in the tissue. So T cells accumulate in mammary gland during the development and during lactation. So to try to understand if the T cells had anything to do with lactation, what he did is actually utilize a scheme in which the genotype of the mother can actually be self-deficient or sufficient, while the genotype of the pups will be unchanged to prevent any form of compounding effect in the offspring. And by doing that, he was able to show that if the mother has no lymphocytes, there is less milk that is actually produced, as you can see here. There's less milk that is actually produced, and the weight of the pups is actually reduced. So this actually supports the idea that the lactation process is actually controlled directly or indirectly by lymphocytes. He then looked at the kind of lymphocyte that were present in the mammary gland. And so, sorry, this is not special for technique, but this is a mammary gland whole mount, when you can see really the depth of the tissue. And you can see the massive accumulation of lymphocytes in the tissue. But what was really surprising is that the kind of T cells that accumulated were all of those expressing Tibet, which is the transcription factors linked to Th1 phenotype that tend to be usually linked to antimicrobial defenses. And it was actually quite surprising because usually developments tend to be more linked to cells such as Th2 or Th17 that will express GATA3 or RNA. It's a bit surprising to have this phenotype in the tissue. So look at to visualize actually what happened in the tissue. You can see here that what happened in the context of an animal that is not pregnant, and this is actually a few lymphocytes that are present within the duct of the mammary gland, and this is a massive accumulation you can see in the context of pregnancy and lactation. And this actually is done by using a Tibet reporter mice, and you can see that if the animal is pregnant and lactate, there is actually an enhanced contact between the epithelial cells and the T cells, and this is actually also linked to the duration of the contact that is increasing over time, pointing out, but not of course proving that there is an association between lactation and T cells. So the next question, of course, to try to understand if those T-bag cells could have anything to do with the lactation process itself, then repeated the same experiment I described before, and found that if the dams, the mothers do not express T-bag, there is less milk produced, and the offspring of the dam that is T-bag deficient have less milk, pointing to this phenotype and this function factor that's responsible for this lactation. It's just a visualization of the transformation of the organ, and you can see here those really the ducts that actually will allow the release of the milk, and you can see that there's a physical change in the absence of T-bag with a less density and less ramification in absence of T-bag in the dams, showing that really there is a physical absence of development of the tissue in absence of this program. So the next question to try to understand which cell subset could be important for this phenomenon. And for that, Dan looked at all the T-bag expressing cells in the mammary gland and was able to show that the one subset that expanded the most in the context of this phenotype was actually the one that is expressing, that is a double negative cells that is expressing NK11. And this is really the subset of cells that expanded the most in the context of lactation and pregnancy. So what do I mean by double negative? It are cells that will not express CD4 and CD8 beta. And this will actually prove to be important for the rest of my talk. The non-immunologists just put that aside. It doesn't really matter. So those cells have this unusual phenotype, and they're treated with non-classical T cells. When Dan looked and compared this population of cells to classical CD8 T cells found in the mammary gland, he found that these cells were actually enriched in factors linked to angiogenesis, response to wounding, but also mammary gland development, really cells that may more be linked to actually transformation of the tissue than really antimicrobial defenses, which pointed to the role and possible role of these cells in lactogenesis. Importantly, these cells were not NKT cells because they do not bind to the CD1 tetramer, which is something that could have been actually be the case because of the expression of NK1. So to try to understand what these cells were about and where they came from, Dan actually started to look at the fate of these cells in the context of their maturation, and was able to see that more mature were the cells, more they express CD8 alpha. So what does this mean? CD8 alpha expression by double negative cells is what actually has been shown before in a very usual subset of cells found in the gut, which are the CD8 alpha IO, which are intraepithelial cells that express this very unusual phenotype. And all cells in the mammary gland seems to actually resemble quite a lot this population of cells by this expression of CD8 alpha. So to look more at the cells and the expression of CD8 alpha over time, he was able to show that in the context of pregnancy, this subset of cells we found acquired the expression of CD8 alpha. And importantly, you can see that in pregnancy, this population of cells really is accumulating over time and really become dominant in the context of lactation. So CD8 alpha expression is something that is really acquired in the context of pregnancy and really sustained and amplified in the context of the lactation process. So furthermore, when Dan looked at the cells, he was able to show that phenotypically, these cells resemble for the most part to the CD8 alpha that had been shown in the gut previously. We have the expression of numerous markers such as Elios and many others. At few exceptions, they express NPM1, that is not the case of the CD8 alpha, and they are highly polyclonal in the HCA, something that contrasts with what had actually been shown with CD8 alpha in the gut. But at the exception of those two features, these cells are really phenotypic of the gut area. So of course, Dan wanted to see if the population of cells we had described was also intraepithelial, really located inside the epithelium. And for that, he needed a surrogate marker because of course Tibet is not only expressed by this population of cells, and he was able to show that enzyme A was actually dominantly and uniquely expressed by this population of cells, giving us the opportunity to visualize this population of cells in situ. And you can see here using in situ imaging of the whole mark of the tissue, then the population of cells we had actually identified were indeed lodged between the epithelial cells and in tight contact with the epithelium of the mammary gland. So at this stage, we could really qualify these cells as intraepithelial cells because they share this localization. And moving forward, we actually decided to call these cells MEL, which will be actually mammary intraepithelial cells. Sorry. And you can see them here well characterized in close contact with the epithelial cell in the tissue. So the next question is to try to understand what was controlling the accumulation of these cells. And it was shown before that the gut IL were highly dependent on the factor IL-15, which is a very ancient cytokine. And in a very similar manner, when you use a mice that is IL-15 deficient, you can see that there is no IL present within the mammary gland. And this actually is interesting because also, once again, you have less milk produced and less growth of the offspring in absence of IL-15. So what this is actually of interest is because the work was done recently by Mimi at studying both in human and mice, showing that actually in a mammary gland of human, there is an accumulation of a cell that resembles a lot the cells we have described that have actually an anti-cancer surveillance phenotype. This actually will actually imply that the cells that we have here uncovered that may be linked to lactation can also share the very fundamental role of surveillance in cancer, something that I think is quite important to further explore. But it also is interesting to also think about the repurposing of the cells in the context of lactation toward another function post-lactation, and may explain the fact that some women tend to actually be protected against cancer in the context of pregnancy. Then if you had lactation, you can actually be less susceptible to breast cancer. The next question we had was to try to understand what would be the origin of these cells. So work that was done by many investigators, including Dan Musa and others, had actually highlighted then the cells that are actually in the gut originate from a cell in the thymus, a very specific precursor. And this precursor here will actually express a specific phenotype and will actually be transfer to the gut where they will become an acquired CD8 alpha phenotype. So what happened during pregnancy? We found that during the pregnancy, it was shown before, although not as studies as everything should be, there's a really remarkable collapse of the thymus. So the thymus really involutes during pregnancy. But what is really, really interesting, and despite this involution of the thymus, is really an enrichment of the precursors of the cells we have actually described that could be actually responsible for this IEL generation. So this is really quite remarkable, particularly in pregnancy, is linked to a shutdown of the classical adaptive immune system, but there is an enrichment of those non-classical lymphocytes, such as those that could be the precursors of the IEL. The next question we had was to try to understand if during pregnancy, these cells are now degraded and go to the mammary gland. To address this question, what we did is to utilize, of course, a setting in which we took those precursors that we purified from the thymus and transferred them in animals that have no lymphocytes that were either not pregnant or pregnant. And what you can see here at the steady state, as shown before, if you transfer these cells in an animal that is non-pregnant, the cells go to the gut. But interestingly, the cells no longer go to the gut in the context of pregnancy, which means that really there is a shutdown of the ability of these precursors to lodge in the gut and to become this IEL. In contrast, when we look in the mammary gland, you can see that those cells really in the non-pregnant mice do not go to the mammary gland, but now those cells are going to the mammary gland in the context of pregnancy. So there is really a striking reshuffling of the cells in the context of pregnancy. They no longer go to the gut, but now go to the mammary gland. And you can see that in the mammary gland, they go and lodge themselves in the epithelial and become the cells that we have actually described. So the next question we had, of course, is to try to understand, to see if really there is a direct connection between the cells and lactation. And for that, what we did is once again to purify this IEL precursor, transfer them in animals, and actually looked at the ability of the animal in the pregnancy to produce milk. And you can see the single transfer of one single subset, this subset we have described in the animal, enhance the milk yield in the context of lactation, and importantly also enhance the number of mammary epithelial cells and neuroepithelial cells. So clearly the transfer of one single cell subset in an animal that have no lymphocyte is sufficient to actually enhance the ability of the animal to produce milk. This is actually what happened, and it's only a couple of markers we have actually looked at in this slide, but there is actually a plethora of them that will change. You can see that actually having this IEL precursor transferred the animal enhanced casein, which is of course one of the main molecules producing milk, and enhance the expression of ACTA2 that is of course important for the contraction of the smooth muscle, and the release of the milk. So there is really a physical potential interaction and an augmentation of key factors responsible for both the quality of the milk and the contraction and release of the milk. There's another image that actually visualizes the potential effect of those IEL precursors in lactation. This is done on whole mount of mammary gland that have been treated with oxytocin allowing the contraction of the milk and the release of the milk. And you can see here, then actually if you do that in an animal that has not received the IEL, there is of course a contraction of the alveoli. And you can see that if you do that in an animal that have received the IEL, there is a physical enhancement of the contraction that will actually be linked to enhanced release of the milk. This is actually shown in the bottom that actually show the enhancement of the amplitude and the contraction and the quantification of the amplitude for enhancement when you have actually presence of IEL. So this IEL really have a physical effect on the mammary gland and in the ability of the cells to actually release milk. So of course, the question that is the most important for this audience is what does it really mean for women? And what does it really mean in the context of the cells present in the milk? So there is a remarkable feature in human milk and in mice milk also, is that the milk is in fact a liquid biopsy that releases a huge amount of cells that are present in the mammary gland. We have actually compared what is present in the mammary gland in the milk, and it's very comparable. And of course, you're going to have release of epithelial cells, macrophages, and many things, but you also have release of T cells that can be present as early as the colostrum. So in collaboration with Florent Ginou, and then also by analyzing a data set that had been actually recently published, we actually looked for a signature of a cell that would resemble the one we have actually identified. And quite remarkably, at a couple of exception of a couple of markers, we're able to identify clusters of cells that is really a phenocopy of the one we have identified in mice, really pointing to the fact that the cytotoxic Tibet expressing program and T cells is actually released actively in the milk and actually, in fact, absorbed by the child. This is another question that we really need to address. What will be the function of the cells when they actually enter the gut environment of the child? So these cells that we have identified in mice actually are present in human milk. And of course, the next question will be to try to understand how this relates, of course, to the milk production and quality in humans. So to finish this first part, what I really wanted to actually highlight is the fact that what we have shown, and what Dan has actually shown, is that there is really, in the context of pregnancy, a remarkable organismal transformation in which the thymus involutes, which actually is in response to hormones. This involution is paradoxically associated with enhancement of the precursor for a very specific subset of lymphocytes that both in the gut and in an IL-15 dependent manner can actually transform in CD8 alpha-alpha. That's a mechanism that we are now finalizing to explore, control the milk synthesis by controlling both the function of the epithelial cells, but also the neuroepithelial cells. And this is actually work that we are now exploring and further exploring in the context of the work we're doing with Kian. So this actually proved that the immune system of the mother, the adaptive immune system of the mother, is actually actively involved in organismal transformation in the physical production of milk. So this observation brings several correlations that I think is of high importance for human health. So knowing that the maternal immune system and potentially inflammatory system can actually impact breast milk quality and quantity means that actually then there could be a change in the context of inflammatory processes or infection in the mother, something that has actually not been explored. And we don't have the data at the moment, but we actually did a proof of principle in animals using Toxoplasma gondii, using tuberculosis. And in every single case, when we have a dam, an animal that has been infected, the quality of the milk is actually changed. The number of the meal is actually changed. The amount of milk is actually altered, and the lipid produced in the milk is actually altered. So this remains, of course, we need now to understand actually what could be the impact on the offspring, but this means an actual infection and inflammation, via potential change in the cells we have actually expressed, can actually transform the quality and the quantity of milk, something that, of course, can have long-term consequences in the offspring. So what we're trying to do now, the work that we're trying to pursue, is to try to develop further this understanding of the quality of the milk in correlation with the cells, and how the number of the function of the cells influence lactation in humans, and how, of course, in the context of more vulnerable trajectories, in the context of infection or malnutrition, this could have actually long-term impact on the offspring. And it is a work that we actually are now developing and would like to develop in collaboration with the Pasteur Network. That is a conglomerate of 33 institutes in the world in which this kind of research can actually be expanded to try to understand a different quality of environment, how genetics, how infection and inflammation can actually influence milk, and then the cell have consequences on the offspring. So on this, I just wanted to thank all the wonderful people that have done the work. Of course, Dan Corral, that is actually a remarkable postdoctoral fellow, that is actually going to be applying for position soon, and many collaborators we had. In particular, I wanted to thank Florent Ginoux, but also Bana Jabri, and many other people in the field have actually helped us, and many collaborators and of course, all our family agencies. And I thank you all for your attention.
We have time for questions. There will be a roving mic. Sarah's put her hand up first.
Yeah, amazing talk, Yasmine. Thank you so much. So this is Sarah Teichmann, and I was just wondering, with the involution during pregnancy, the thymic involution, and the development of the more of these interesting kind of CD8 alpha intraepithelial lymphocytes. I guess one of my questions was whether this is kind of going more to a sort of fetal-like thymus, because in early thymic development you get less of the classical kind of CD4 and CD8 sort of mature cells and more innate like CD8 alpha, gamma delta, and so on. So that was one question. And then if I may, the second one was whether there's any change in kind of chemokine receptor, you know, for the trafficking to the mammary gland versus the gut, and this sort of egress from the thymus and so on. So I'm curious about those two sorts of, one is kind of a bit more high level, the other one is a bit more molecular. Yeah.
You're absolutely correct. And I think this is a fascinating observation, which is the fact that pregnancy is somehow reversal to a more primitive form of immune system. And this is something we see in many different parts of the body, in which the non-classical immunity, all this immunity that we know tend to be more dominant in early life, now come back to the rescue. We all know that actually during pregnancy, we do not want to have the strongest adaptive classic immune system because it could also mean responses toward the fetus. But you still need to maintain an immune system. I mean, women are not immunodeficient in pregnancy despite the strange narrative of the past that we are immunosuppressed. We're not immunosuppressed. Women are in fact changing their immune system toward a more non-classical, more broad reactive immune system. And this is exactly what you said. What we find is in the context of pregnancy that the decrease of the timings is linked to this more enrichment, more generally non-classical, not just the subset. All the other non-classical become dominant. So it's a fascinating shift and organismal transformation toward maybe what we refer as a more fetal, but I will refer as a more primitive form of immune system that evolutionary is in fact was actually more dominated by non-classical responses. The second one is you are correct. There is definitely a shift in the chemokine production, and the various chemokines that have been shown to be important for the eye of recruitment become highly dominant within the mammary gland. And we think this is also one of the mechanisms, but we're not sure it's the only one, because we don't see a shutdown of the chemokine in the gut. So we see the enrichment of the chemokine in the mammary gland, but no shutdown of the gut. So this could be one of the factors attracting, but there's still something else. So I think there is a dual heat that allow the cells to accumulate in the tissue, that allow them to lodge themselves in the epithelium. So we now look at that. Of course, there is enhancement.
Okay. No, that makes sense. Thank you so much, and get well soon.
Hi, Yasmine. This is Musa. Really beautiful study, beautiful talk. I have a really brief question, which is, it's been shown, I think in humans, that non-pregnant women can also lactate. So what and how is this mechanism being harnessed to make meals in non-pregnant women in an oxytocin-dependent manner? I saw that oxytocin data that you showed more for the alveolar part, but how does that happen in humans, and can it be replicated in mice?
I mean, so we haven't, in our case, we cannot actually make an animal lactate just by transferring the cells. So clearly there is other factors. But you could even like a baby can actually sometimes have a lactation, which is, this can happen. And you could imagine that actually those IL, if they were to go in the child, could potentially be responsible for this kind of transient lactation, the rich milk, I think this is called. And for the woman that is actually pregnant, it could be very likely possible, and in some circumstances, a deviation of accumulation of the cells that go chronically from the GI tract toward the mammary gland to contribute. In fact, it could be fascinating to see if women that are non-pregnant are able to lactate, to actually just look at the milk and see if the cells are present. That could be a way by which it could actually be associated.
Thanks, Yasmine. I've got two questions, actually. One, given your role now as the president of the Pasteur and the kind of difficulties actually in getting funding for research into kind of like women and reproductive health, I wondered what your thoughts are on how we can actually change this. That would be kind of like the first more global question.
I think my first question. So I think we should shout loud and clear. Then this is no longer negotiable, and I think we should just make sure that this research is actually done. When I had started to actually this journey on working on pregnancy a few years ago, and we were just looking for data sets telling me, okay, what is the immune system of a pregnant woman? And realizing that we didn't know that actually made me angry. So I really think it's time for us to become fierce advocates. So one of the things I have been doing at Pasteur Institute is this become now one of these, in my strategic plan, one of the priority of the Institute Pasteur. That's going to be one of the scientific priority of the Institute, working on maternal child diet. So I think that's what we need to do to make sure that we advocate, do it ourselves, take do this science, and make sure that when you have leadership position, make sure that this is actually part of our roadmap. And also, we need to also make sure that our deciders are informed, and that's why I'm spending a lot of time doing advocacy and giving talks and going to talk to governments to make sure that this is no longer neglected. If you think about the field of immunology, we are probably 20 years behind in terms of trying to understand pregnancy. In terms, we don't even know enough about the immune system of women and men. Let's not even talk about pregnancy. So I think this is an unacceptable state of the world, and something that we need to become, I think we need to become angry and activist. That's my point.
And I guess, how do we convince industry that there is actually a huge return of investment? Because there is no, it's not on their radar as well as, governments.
And it's a mistake, because I think disease, etiology of disease, treatment of disease, response to vaccine. I don't know, can you see me? I'm still a bit frozen. Can you hear me?
Yeah, we can hear you, but we couldn't, you were frozen, but
we
we can hear you.
Yeah, I mean, treatment to vaccine then is different. We had a paper recently here, at least, showing that this, response to immunity of vaccine in the skin is way more reactive in women, which we know, and which means that vaccine dose can actually be decreased. So why on earth do we not integrate this fundamental knowledge in every single biology we do? And I think it's of great interest for industry to just make sure that they can develop tailored approaches to the treatment, prevention of disease, or even response to vaccine.
Great, thank you. On that note, that was a fantastic lecture, as always. And, thanks so much for actually kind of like speaking despite your illness and pain. So we really appreciate that, and we send you our best wishes and get well soon from this hall. Thank you so much once again, Yasmine, for joining us today this morning, and we wish you well. We now move on to the next session, which is an update from the Diversity Task Force, and that will be moderated by Shyam Prabhakar. Oh, Musa now. Okay, sorry. Musa Mhlanga, who is going to be the moderator. My apologies.
Oh, that's okay. Sophia, go ahead.
Hi, thank you for joining us for this session, and we're going to start right away with Sophia, who's going to give us some feedback from the DTF, or Diversity Task Force.
So, good morning, everybody. I am honored to speak first on behalf of the co-chairs of the Human Diversity Task Force through the HCA. So I'll get us going. One of the things that we think is important to highlight is that there is variation across disease and incidence and mortality across the world. And here are two examples for breast and cervical cancer. We posit that, of course, there is a variation in the burden of disease due to access to care. But we also see, for example, in the context of breast cancer, that there are populations, there are populations like in Europe where you have high incidence of breast cancer, and also in the Caribbean. And we know that there are differences in molecular phenotypes of these tumors, and also the age at onset in these tumors. So we know that geographic variation is playing a role in addition to genomic ancestry in disease etiology. When we look at data that's been published with polygenic risk scores, well, here, the reference panel here is the European ancestry population. We see that these polygenic risk scores are not great predictors when applied to different populations, and you can see here. Furthermore, I'm looking at single cell data, and this is data from IDA. We see that in data that is recently published, that genomic ancestry plays a major role in describing subcellular variation in immune cells. And further, when we look at EQTLs, we see that there's an abundance of population-specific functional variants impacting cell site-specific expression, which can only be identified through studying diverse populations. And you can see that in blue here, that the IDA-specific EQTLs represent about 25% compared to a lower proportion in the GTEx-only data. Additional research has been published looking at disease-specific cell abundance. In this example on the left, we see that there are differences in the abundance of CD4 naive T cells in individuals of Asian ancestry compared to European ancestry. And most recently, Harry published his work looking at different proportions of breast epithelium, healthy breast epithelium, across multiple populations within the United States. And he shows that, in particular, the LASP cells, which are the alveolar progenitor cells, have different abundance in people of Native ancestry and Native American ancestry indigenous populations. So we have a history of studying populations globally. This is now a 2-year-old study showing that the GWAS studies have had an abundance of European ancestry, which is not in proportion to the global population. But in addition, we see that there has been an increase over the past 10 years, an exponential increase in the number of studies, but not concomitant increase in the populations that are underrepresented. This is data from the cell by gene census that Kian pulled down, and it shows that the proportion of people of non-European ancestry, again, is low. And when we look at the data from just one year, 12 months ago, from then to now, we see that there's an exponential increase, a 40% increase in the number of donors, but that proportion or the increase is not represented in people of non-European ancestry. So there's been work afoot to improve those numbers, and we expect that to be improved through the intentional funding of groups and research groups by Chan-Zuckerberg Initiative, through the Ancestry Network, where they have supported a lot of studies across different tissue types and cell types across different populations. And so we expect that number to be increased in terms of those people of non-European ancestry. So this is the organizational chart of HCA, and through the equity working group, there was a task force that was put together. And this task force is now representative of people across different populations who are leaders in their spaces. We thank them for their participation, contribution, specifically Samira, Robert, Gideon, Yusuf, Vinicius, Pompan, Drew, Elisa, Yukinori, and Patricia. And in addition, we had observers from the Equity Working Group and other groups within CZI. We also consulted with different external partners to ensure that what we were doing was in alignment with other goals and recommendations in other entities internationally. And of course, we had extra support from the HCA staff, John, Christine, Esther, Lucia, and Tracy. So thank you very much. So we came up with 4 terms of reference. Term 1 is focused on the ethical and scientifically appropriate process for engaging underrepresented populations in sampling and monitoring efforts. Term 2 is the use of diversity-related metadata across all HCA data platforms. Term 3 is on metrics and resolutions for tracking genetic and geographical diversity in HCA. And term 4 is the HCA goals for genetic and geographic diversity, for which my co-chairs will expand on. I just want to talk about term 1, which is the ethical component, where we want to ensure that the work that we're doing is maximizing benefit. We want to recommend close consultation and engagement and collaboration of leaders local researchers across our communities that we want to increase representation of, in addition to capacity building. So we're not doing helicopter science. Obviously, it's an expectation that we get IRB or research ethics approval in the local context, and that we want to improve the inclusion of rural, marginalized, and or underrepresented communities with a focus of benefit sharing to address a local unmet need.
Hello. Thank you, Sophia. So, buongiorno, everyone. My name is Kian. And this next session, this next part of our Diversity Task Force recommendations talk, I'll be speaking about some of our recommendations on human diversity metadata, as well as thinking about our metrics for and reporting on human diversity in the Human Cell Atlas. So I'm going to start with a schematic over here, or two schematics. So the one on the left is actually a representation of some of the reference populations in the 1,000 Genomes Project. And the one on the right is actually some of the reference populations in the Human Genome Diversity Project, or HGDP for short. So one of the things you might notice is that, for example, the Southeast Asian region over here, which I've circled in ellipses, and that's where I'm from Singapore, that's actually very sparsely represented in these commonly used genetic variation references. So for example, Indonesia, a country of almost 300 million people, is not at all represented in the 1,000 Genomes Project, and that's actually probably the 4th or the 5th largest country in the world by population size. So really, if we try and measure or represent donors in these populations, so in Indonesia and Malaysia, in Thailand, or elsewhere in the Southeast Asian region, and think about genetic similarity to reference populations. And I think we're actually missing out on quite a lot in terms of representing the donors in their proper context. So really, throughout our Diversity Task Force discussions, we came together to think about what is really a suite, an array of metadata fields that help to best represent or help to better represent human diversity, as well as fields that are relevant for thinking about correlation with physiology and pathophysiology. So you heard from Sophia in the introduction just now about some of these dimensions of human diversity, and we've narrowed down a list of categories that we think are good or are proxies for factors that can affect human health, healthy baselines, as well as these risk of phenotypes. So in addition to genetic ancestry that I alluded to earlier, in terms of thinking about genetic similarity, for example, we can also think about self-reported ethnicity, language, geography, age, sex and gender, as well as environmental and lifestyle factors, including things that we think about that are social determinants of health. So one thing to note is that one of the recommendations we'd like to make is that this human diversity metadata should be embedded in every single bionetwork, so all of the 18 bionetworks in the Human Cell Atlas, as well as all versions of the reference atlases, because it's really important for understanding donor context and also being able to represent covariates for analyzing possible confounding factors or factors of interest for understanding the impact of human diversity on biological phenotypes. You heard a lot yesterday from Chloe over there, who spoke about integration and about the Tier 1 metadata. So in the context of that schematic, this is actually fitting into the Tier 2 human diversity metadata, alongside all of the bionetwork-specific fields that you heard about in the presentations yesterday. So over here, on the left, I've borrowed this logo from a recent talk I heard from Jay Tyagaraja from Boston. So metadata is a love note to the future, and if you collect really that comprehensive suite of metadata now, you're bound to be able to reap the rewards, 10 years down the line. And yesterday you heard from Nicole Savanzo's keynote talk about that wealth of metadata and genes and health, and hopefully in the future, UK Biobank being able to link the multitude of disease phenotypes, of measurements, of physiological measurements and metadata to all these single-cell measurements. So really population-scale genomics and population-scale single-cell genomics. However, from the perspective of someone, say a study participant filling out these pages upon pages of questionnaires, or to the data contributors in the audience over here, looking at reams and reams of spreadsheet, this can seem quite intimidating to fill in, and if you have a huge list of metadata fields you're collecting, the accuracy might go down, and you might not be as motivated to perhaps fill in 100 fields. So what we've done is really, in the task force, to think about priority tiers for metadata. So what is the balance between the comprehensiveness of metadata collection that can inform rich downstream analyses versus something that's perhaps pragmatic to implement retrospectively for many studies, as well as going forward to recommend in study design for all these population skill studies and other cohorts going forward. So in our metadata priority tiers, we've got a prioritize, so that's our highest metadata priority tier, as well as recommended metadata. And this is in the current HCA spreadsheet that's going to be circulated to all of the bionetworks. We've also got some supplementary metadata. So this is in our specifications. We probably don't expect most of this to be represented in existing studies, but we hope that this will inform future study design. And finally, at the back end, we recommend that continuous measures of genetic ancestry and genetic sex are actually being inferred in order to represent this in a continuous fashion to complement some of the categorical, self-reported descriptors that you see. So in thinking about illustrations for how this schematic will look like, so over here I'm showing the various categories that I spoke about. So over here, this list is actually that of the prioritized metadata that we hope will go out to every single bionetwork. So you've got, for example, self-reported ethnicity and language with multiple options possible that capture some facets that may not be represented if you think about genetic similarity analyses. So you've got self-reported gender identity and donor current residence, and as well as certain lifestyle and environmental basic metadata fields that we hope will be easily or more easily captured across sites. So things like BMI and collection site location, we think that these are perhaps more pragmatic to collect. And collection site location can serve as a proxy for environmental factors. So if you think about the air pollution index, or you think about the incidence of malaria or infectious diseases globally, so these sort of geospatial proxies can be representative of some of the environmental exposures that our donors have been exposed to. So over here, in this next slide, what I'm overlaying is actually a series of metadata fields that are colored in blue. So these are the next tier, recommended metadata. So we think this would be challenging to collect, but still highly informative. So thinking about, donor place of birth, about your parents' self-reported ethnicity, and if you speak to Sophia, you hear anecdotes about how actually people convert their self-reported ethnicity in terms of in certain regions of the world when they actually undergo marriage. So being able to report your parents' self-reported ethnicity as well as your mother and father tongues can actually capture some of these, measures that may not be so apparent when you're just reporting on your current state. So to sort of sum up this metadata, human diversity metadata recommendations from our task force, so we've got a series of priority tiers that's depicted in this schematic on the left, and for each of these fields we've got detailed instructions and guidelines to data contributors, and we hope that this will go out imminently soon with some of the tier 2 metadata as well as the standard operating procedure protocol documents that Chloe actually showed QR codes to yesterday. So these will be available, we hope, soon on the HCA resources page in time to come. So the next related term of reference that I'd like to talk about is really thinking about metrics for human diversity in the Human Cell Atlas, and how do we report on these metrics? So you saw from Aviv's and Sarah's slide yesterday that we've now got more than 3,000 members over 101 countries, and the HCA is actively tracking some of these membership metrics. So in addition to membership, what we hope is that for each reference atlas, the HCA will be able to track and report on the geographical diversity of sample collection sites and sample numbers. So analogous to the number of members across the various inhabited continents in the HCA, we would like to think about the number of samples per country and per continent, as well as the proportion of countries that's represented across in each continent in each reference data set. In addition to some of these metrics for tracking human diversity, we're thinking about ways of depicting the extent of diversification in each of these reference atlases. So on the left over here, I've got a schematic of populations or countries that are actually represented in one of the first assembling the first draft of the Human Cell Atlas Genetic Diversity Network, Human Diversity Atlas. But however, this country-level representation doesn't quite capture environmental diversity. So since we're in Italy, we're in the Tuscany, Milan region, it's going to be very different from Sicily, for example, in terms of the environmental exposures. So I've got an example on the right where we're recommending perhaps geographic heat maps based on some of the GPS coordinates of collection sites that will be able to reflect some of the environmental diversity, thinking about the level of states or perhaps some more granular resolutions. As we think about some of the goals for human diversity in the HCA that Shyam will talk about in the next section. So one of the things I would like to add as a caveat over here, and thinking in the context of Sophia's, what Sophia said about engaging ethically engaging with underrepresented populations, is that especially for population groups that are fewer in number and where the risk of re-identification is likely, we would like to, I think, caveat or caution data contributors to think about blurring or binning strategies so that we are able to represent these underrepresented populations, but also be wary to avoid, to maximize benefit and minimize risk, such as donor re-identification for these population groups. In addition to thinking about geographic heat maps or overlays on the world map, so for each reference atlas, we will also like to recommend summaries of self-reported ethnicities. So think the top right pie chart over there, that's taking from really a US-centric point of view, where in the US, in terms of ethnicity, people think about Hispanic, non-Hispanic from some of the traditional census surveys that are being done. And now on the left, that's actually how, I think in the US, we think about race descriptors, so Black or African American or white. But there's actually differences globally in how we can think about self-reported ethnicity, tribe, and race. So in the bottom, we've got some examples from the African Caribbean network in Kenya and Nigeria, where there's this really diverse array of self-reported ethnicities, races, and tribes. So I think being able to report this and take into consideration some of the different contexts in which race, ethnicity, tribe, reporting is being done by different countries. I think this will help to show the extent of diversity in each of the HCA reference atlases. And finally, I think the last sort of reporting or illustration I'm going to show in terms of thinking about reporting of human diversity in the Human Atlas. So here's another example from the African Caribbean Network. So Sophia and Alex, who's actually in the audience here, has generated this projection of some of the cohorts in Miami, Nigeria, and Bahamas against superpopulations in commonly used genetic reference databases. So for each reference atlas, because we're going to be able to get the FASTQ files through managed access, we want to recommend really summaries of donor genetic ancestry, so inferred perhaps from the single-cell data themselves in order to be able to capture or reflect diversification in the atlas. And I want to end on one thought, that if we're able to go beyond these reference landmark populations to fill in all of the gaps, so for each of the reference atlases, being able to have population groups that were not represented in reference atlases, in reference genomic data sets, present in single-cell data sets, I think that's really testament to the ability of the HCA to achieve diversification and also global benefit. So I'm going to hand over to Shyam, who talk about some of the HCA goals for diversity.
Thanks, Kian. Thanks, Sophia, as well. So I hope you've gotten the sense that it's extremely important in the Human Cell Atlas for us to study diversity, study human diversity. As Sophia mentioned, disease risk is different when we get it across the world. When we get the disease, the prognosis is often different, disease processes are different, response to drugs is different. And so clearly we cannot do all of our science in only some parts of the world and only some subsection of humanity, right? So that's the motivation part. And then Kian Hong has spoken about how we get there. When we say diversity, what do we mean? What are the major axes of diversity? So ethnicity, ancestry, geography, sex, age. We want to address all of those, and it's incomplete, of course. We want a lot of environmental metadata. But the metadata schema that we've come up with in all these discussions in Human Cell Atlas, in the Diversity Task Force specifically, tries to do some kind of compromise between, as Kian Hong was saying, the completeness. We want to know everything as researchers, and the practicality, which is what are the most important things we should focus on if we want to reduce the burden on the submitter, right? Now, so that's great. So we have motivation, we have some methodology, but the second part of what Kian Hong presented is really the key, right? How do we avoid the fate of all these other consortia that also meant well, that also understood very strongly that it's scientifically and ethically and equitably important to study diversity, but yet they plateaued at a very small fraction of diversity over 10 years, right? Can we do something differently? And the one thing we converged on was setting metrics and goals, okay, to hold ourselves accountable. And that's what I'm going to talk about now. We're sticking our necks out here and committing ourselves to achieving some targets in terms of diversity. Thank you. So primarily, we want goals for sex balance, geographical diversity. We want goals for ancestral diversity, also I'll come to that, and environmental diversity, right? Why have we not specifically included numerical targets for genetic diversity? It's not obvious how you would do that. So the obvious thing is to say, okay, European ancestry, African ancestry, then Asia, sub-ancestries, and so on. But really, the field is moving away from this concept of ancestry as a discrete thing, because it causes all kinds of problems. Right? What we know biologically is that ancestry is complex. Genetic ancestry, for sure, is a continuum. It's rarely discrete categories, right? So we want to stick with the continuum view of ancestry, avoid labeling people genetically. And if you do that, it's not so obvious how you set quantitative targets for ancestry. And so instead, we took the route of setting quantitative targets for geographical diversity. Geography is also continuous, and ancestry is continuous mostly because geography is continuous, okay? But at least geography is demarcated, there are national boundaries. So you can, in a very straightforward way, discretize and categorize geography. And so we set geographical diversity goals, then we automatically get a large part of our genetic ancestry diversity goals met, right? And so that's how we quantified that. So for sex balance, I think it's fairly simple. We want at least 40% male, 40% female. This is the relatively easy part. Unless you're studying, like Yasmine presented just now, biology of lactation or something like that, then, obviously we're not going to insist on 40% male in your cohort. But for the most part, we say, okay, at least 40% male, 40% female in each reference atlas. And then the harder part is geographical diversity. I'll come to that. What are our goals over there? For environmental diversity, again, quite challenging. Primarily because it's very hard to collect harmonized metadata on environmental variables. And the UK Biobank has done a fantastic job of that. There are other consortia, even in the HCA, that are collecting very detailed environmental information. People like Sophia are very well engaged with the communities and knows them personally, the people she works with, and they are very motivated to contribute as much metadata as they can. Not everybody in the world has that ability. And so at least at the bare minimum, what we want to collect for environmental diversity is urban versus rural. And you can say there's suburban also, and how do you define urban and so on. But we do want a diversity of population densities in the HCA. Okay? And there are people funded by CZI Ancestry Networks collecting samples from very remote areas in Asia, Africa, South America, all over the world. And I'll come to quantitatively our metrics for geographical diversity, okay? Because that's perhaps the most challenging. The key thing we want to achieve here is version-to-version improvements. To avoid plateauing, we're committing that your version 2 of your atlas should be more diverse than your version 1 of the atlas. It was not mentioned as one of the criteria for an atlas because we're still in the version 1 atlases mostly, but we hope that that will be one of the criteria, right? For all these kinds of diversity. And then we're talking, everything I've mentioned just now is about diversity of healthy atlases, but you've heard the buzz throughout this meeting that disease applications are important, and I'll come to that in the last slide. So that's another goal, that we want to use our healthy reference atlases to derive insights into disease for diagnostics, for therapeutics, and that's how we will turn our research into something that can benefit communities worldwide. Okay, that's our final diversity goal. This thing is very new. We do not have quantitative goals for this. We're just starting the conversation over here in Milan. But we hope that this will be a major new part of the Human Cell Atlas. So goals for geographical diversity, conveniently, we already have an HCA metrics dashboard. HCA has 101 countries, and there'll be some kind of baseline for our geographical diversity goals. And so here it is. We start by reference to the blue bar, which is how many countries have PBMC data in the CZI cell by gene corpus, right? Why PBMCs? Because PBMCs are perhaps the easiest data sample type to access. And so PBMC data are more diverse than the other data types in the HCA, right? We're already at around 11 or 12 countries for PBMC data sets submitted to the HCA data repository and to CZS, the matrix store, cell by gene. What do we want to do in 5 years? We made a very rough calculation. Kian Hong showed that slide earlier. You have around 40% year-on-year expansion in the size of the data set. So if you compound interest that over 5 years, you get roughly 5-fold expansion over 5 years. So we said that we want to get, but that's a 5-fold expansion in the number of cells, number of donors, okay? Not so straightforward to get a 5-fold expansion in the number of countries represented in the data repository. But that's our goal. We decide that we will be aggressive. And it's not like 50 countries in Europe, right? So I don't know if there are 50 countries in Europe. But we want these countries to be uniformly distributed across the continents globally. So this 50 countries means a minimum number of so many countries per continent. That's for PBMCs. We expect a 5-fold expansion here. Then there are some intermediate sample types, say nasal swabs and skin, that are relatively still accessible, minimally invasive. And for that, we're setting targets that are almost as aggressive as for PBMC, so 30 countries in 5 years. And then at least 3 other bionetworks, we can't prejudge today, should reach the same level where PBMCs are today, which is around 12 countries in 5 years, right? That's a 5-year goal. In 10 years, we're ambitious. We're saying all bionetworks should hit 50 countries worldwide, uniformly distributed across the continents by then. I think it's achievable, and looking forward to all of your contribution to make that happen. I shouldn't say I think it's achievable. The Diversity Task Force as a whole has stuck its neck out and said that we can do this. And lastly, I'll come back to the point of disease and emphasize the mission statement of Human Cell Atlas that Sarah and Aviv brought up way at the beginning of this meeting, which is we want to create comprehensive reference maps. So the word comprehensive is important. So comprehensive means it has to be diverse. Human cells, fundamental units of life, as a basis for understanding human health, for sure. And you want to profile diverse cohorts to understand human health. You can't understand human health by profiling cohorts just in one part of the world. And then the second part of the mission statement is for applying all of these healthy atlases as a basis of diagnosing, monitoring, and treating disease. Right? The time is ripe to launch this part of it, and that's why even way in the beginning, a disease focus was envisaged, or disease applications, you can say, were envisaged. And they're quite relevant for us from a diversity perspective as well. And that's why it was a strong recommendation of the Diversity Task Force that HCA prioritize disease applications. Why is that? One very simple reason: you want precision medicine and a medicine that helps everyone. And that means that you need healthy baselines and your disease risk, your pathophysiology, all of it. You want the science to understand how diversity impacts all of that. And a more practical consideration as well. Many countries in the world, or many researchers all over the world, simply cannot participate fully if we're only talking about healthy atlases. And that's because the communities they work with want to see the benefit of their research. That's one. Then their funding agencies have a priority for disease application. And that's again true with many researchers, many countries we've spoken to. put it in Singapore, where I come from, first world country, but still the focus is on disease studies, right? So if we want a big tent, want to bring everyone in, we have to focus on what matters to them, what matters to their communities, right? So that's one major motivation. And that means showing benefit for those scientific communities and then building the basis for precision medicine worldwide. Right? How do we prioritize diseases? We do not want to do that in a top-down manner, and we cannot tackle all diseases for sure. But we want to prioritize a handful of diseases that the HCA puts its weight behind. At least that's the hope. Nothing concrete yet. But as we go down this road, we want to consider feedback from all of the HCA regional networks. What are their key priorities? Local needs for locally relevant or endemic diseases, as well as global disease burden. I'll give some examples of that. So here, if you just look globally in terms of disability-adjusted life years, just pure numerical metric, okay? What diseases should we prioritize? And I'm not saying this is our only metric, but this has to be one of our considerations if we want to achieve benefit globally, right? So COVID-19, of course, was number one recently, coming down, and heart disease, stroke, respiratory infections, and so on. Definitely, we need to consider these in our planning and our prioritization. But of course, global disease burden and local disease burden are not the same. And so we just picked the example of Pakistan, more than 200 million people. It's a very large country. And you can see that preterm birth complications, because it happens early, very early in the developmental cycle, is number one, actually. But you won't see it in the okay, you see it as number 5 in global disease burden, okay? But, so this is the balance we want to strike between local and global factors, and also capability, let's be frank, what funders are interested in. And then competitiveness, meaning what are the efforts that are already ongoing, so how can we be complementary? All right, so that's a disease focus. I want to come back to the start of this diversity task force, which actually was started in a conversation with Musa. So Musa is the person who said, we need to do something concrete. We need to set up a committee that will actively lay out some strategies and some goals for this. And now it's up to you, all of you, to make this happen. Please join the Genetic Diversity Network. You can do so by emailing this email address, and diversify the HCA. So incorporate human diversity in your study design that includes metadata, metadata, metadata. Without that, you cannot study this. And then help to drive disease applications and submit your data to the HCA data repository. Right. Lastly, I just want to thank a few more people who are part of all these conversations. So Aviv and Sarah for supporting this wholeheartedly from the start and giving us feedback in Zoom calls and in meetings. Chloe and Holger, who have given a lot of feedback as well and guidance to the Diversity Task Force, and then all the people Sophia mentioned as well. Thank you.
So we have about 15 minutes or so before the break, and I guess this is an opportunity for people from the audience and also for our panelists to answer a few questions about not only the origins of this and what's been motivating it, but also I think we should really pay attention to the difference I think the HCA has been in this effort, which is from the beginning, and this has come from, I guess, I shouldn't forget the people who were there in the room at the very beginning, like Eric Lander, but also Aviv and Sarah, really diversifying and making this atlas equitable from the point of view of a healthy atlas has been a primary goal. And last year, Aviv brought up, I think, something really important in her keynote talk, which was that the atlas needs in its next phase to become far more diversified. And that was really what motivated us to try to drive to create this very focused diversity task force. So I guess my first question is, we know there's a lot of infrastructural challenges, regulatory challenges. There's a huge amount of unevenness in the technological capabilities around the world. And to be frank, also financial capabilities around the world in terms of achieving these ambitious goals. So these are very core challenges. How would, I don't know who wants to take this question, but how have you thought about trying to assist and overcome some of these? Maybe I'll start with you, Shyam, since you were just on stage. So go ahead, tell us your thoughts.
Thanks, Musa. You outlined very well some of the challenges that have limited diversity in other consortia. Primarily, if you can, it's not only about funding, but funding is a major part. If your national funding agency is not enthused about the work, it does not align with their priorities, then it's hard to get funding. But of course, even if your national funding agency is enthused about it, some of these projects require funding on the level that you cannot get with local grants in certain countries. So that's a challenge. I have to, all the foundations that have stepped in to make this work possible on Human Cell Atlas, definitely huge thanks to them. CZI comes to mind for sure, Sangha also in supporting such research. But funding is not, we need as a community to organize some funding mechanisms to support this kind of work. That will be essential for overcoming this challenge and increasing diversity. Right? NIH is not going to fund all of this. Funding is not the only thing. As you mentioned, training and infrastructure are the other factors. We all know, even among well-funded researchers, if you get money to do something that you don't have experience in, you probably won't deliver, right? And anyway, the peer reviewer should not give you that money. So training and infrastructure is extremely important. And here, I want to highlight the work that Alex has been doing, training, and you and others, to build the capability. And those two go hand in hand. So for example, CZI Ancestry Network's funding, it increased the capability and expanded the talent pool in all of these countries and allowed them to generate pilot data, which helped them access local grants, which allowed them to hire even more people and so on. So it's a virtuous cycle over there, right? And the last is infrastructure, where again, you have to seed the infrastructure with some of these pilot projects funded somehow, and then that motor can keep on running and supporting a whole bunch of other projects. So that's a goal, but I hope maybe Sophia can comment on how she's addressed some of these issues.
Yeah, so in addition to what you just said, Shyam, it's also about democratizing the data that is generated, so that for those spaces that are not necessarily interested in building that type of infrastructure, again, the motivation of why they're participating most of the times is because it's a disease-focused space. It's also providing access to data sharing, to being transparent with what it is you're doing, why it is you're doing it. And then I would say the move into spatial has also opened up the opportunity for more participation because of the use and the availability of FFP tissues across everywhere. Of course, there's still optimization to be done, but in general, that's accessible, and so that decreases the ability for them to participate, as with single cell and having the infrastructure in place for even snap freezing tissue. Yeah, it's an ongoing conversation all the time, and again, justifying why it is that we are going into these specific spaces that you have to explain what the single cell is, why it's important, and then them telling you that really they want training for other things, and making sure that it's bidirectional in the relationships that are being built, that there's benefit for both.
Maybe one quick thing I'll just add is that really I think outreach, so drawing an anecdote from HCA Asia, Ponpan and Varadom, our IDA colleagues, organized a very well-received HCA computational workshop that was supported by Alex Shalek and members of the HCA staff. So I think there were 13 countries across Asia Pacific represented there. So going out there, engaging with trainees, being able to share computational experimental techniques, get people excited about the work. I think that's really a concrete thing. So as part of our task force, we spoke with a lot of people, and also with our task force members and observers. People like the road shows that I think HCA used to run. So being able to bring this to diverse regions globally, I think that's an actionable next step to help with some of the capacity building, thinking about infrastructure, getting people excited that Musa asked about.
Yes. We have a question from the audience here. Or is that an online question?
Online question.
Online question.
It's from Fernam Iqbal. Hi, can the speakers give us an idea that how, e.g., people working in developing countries can contribute towards the Atlas projects, given we're provided by with little funds for the basic research, and such advanced research is not even in the schedules? Can we contribute by providing samples that are missing in the West?
Yes.
Yes.
Please join.
Yeah.
And yeah.
And that's the whole point about democratizing the data, training on how to analyze the data. So even if you can't generate the data, that there's access to having the data back and contributing beyond just contribution of samples, so that they stay engaged.
So they should email genediversity@humancellatlas.org.
Thank you for remembering the email address.
Thank you. And we really like this to be a two-way conversation, so it shouldn't be unidirectional.
We have a question at the back.
Good morning, all. First of all, I want to thanks for this talk. And on top of the first question that you talked about how to be sure. Actually, you talked about the funding aspect, but one thing that I'm thinking now is especially about the Middle East, and you all actually consider this part of the world, I think, really good, because you actually consider one day to this part of the world. So I want to know how you will be sure that these improvements can be applied in these countries, because I think it's not that much easy to be sure that all of the people across the world can access to these improvements, because your goal is to be effective also for the people who lives in the rural parts of the cities and countries. So I think sometimes talking about something is easier than just doing that in real world. And also, these meetings all be held in English. So, for example, I'm talking in Persian, so my people maybe can't reach to this news online. Or we know that these days, Instagram, Twitter, and these stuff are really well known. But so, if it's in English, so I can't access to it. So I think normal people may need some people who can speak in their language sometimes to actually be aware of these improvements, and then ask the country, so they are doing something about these things, so please, we are requiring these improvements in our country too, something like this. I don't know if I just explain it well. So yeah, that's it.
Which one of you would like to make a brief answer to that question?
I can. So in general, the first term of reference that we indicated was engagement, ethical engagement, and that means with intention. So as we build these diverse atlases, the goal is not to do it from a Euro or American-centric space, that we're working in collaboration with the scientists, the physicians, the community workers in the countries. And so that means that you get to translate what we are doing and engage with your community in best ways. We also work with our collaborators, at least I speak from my network, work with deans, work with heads of departments, and also with government, so that it facilitates and they understand why we're doing what we're doing, and we're not just like coming in, doing something and leaving. And so that builds their capacity to be able to communicate in any language that's relevant to the specific community, so that they understand the benefit. That's the part about the benefit sharing. And so it goes beyond basic scientists. It goes with health workers, linguists, historians, actually, to give us context of why it is we're doing what we're doing in these particular specific regions, for example. So it has to be like engagement across multiple disciplines. And so you're asking those questions, and I'm thinking, yes, like this is meant to be a full engagement so that we can actually share that benefit to the local populations, but it's done in collaboration.
Thanks, Sophia. Sorry, we don't have much time, so we'll take, I guess, one or two more questions. Brief question. Partha?
I have a question. I really want to share my experience, but very briefly. Most of from what we know from past genetic studies, the most diverse parts of the world are also most challenged to be able to participate in these kinds of studies. And like the entire group pointed out, that bringing in diversity is so important here. I just want to mention a few things, again, from my own personal experience collaborating with many international consortia in the past. The one thing that needs to be done by, again, I'm being very prescriptive, take it or leave it. Funds are required to implement these prescriptions, but I will be prescriptive. One of the things that the HCA leadership needs to know, and I have personally benefited from Broad Institute, from Wellcome Trust Sanger Institute, Aviv and Sarah, is to get people from these parts of the world, go and get them to their own institutes and train them. That's the first thing to be done. The second thing to be done is a reverse direction. That is, come out of your shelves and organize these workshops in the countries of the world where there is a lot of diversity and you want them to participate, because there is no appropriate technological wherewithal, no appropriate scientific wherewithal to be able to participate in projects such as single-cell projects. So even organizing these workshops will need money, so a certain proportion of money needs to be devoted for bringing in people from these kinds of countries into HCA and to be able to contribute. The second point that I want to make is that, and this has been said multiple times here this morning, that in these parts of the world, it's also very difficult to generate funding from national agencies for two reasons. The national agencies themselves are not, the decision makers are not completely aware of the goals of these kinds of projects and what translation might these bring in. May not be in the short term, but maybe in the longish term. So the leadership of HCA must come to these countries, engage with the national funding agencies and the leaders of the funding agencies. And the International Cancer Genome Consortium had done that very profitably. And I would say that the HCA leadership must also do that. And if they do that, then probably there will be some money that can be generated from these, from the national funding agencies, and that will increase our ability to join the Human Cell Atlas and profitably contribute to the Human Cell Atlas. I'll stop there.
Thank you, Partha. Just behind you, please, first. And please, very brief, we don't have much time.
Yeah, so very briefly, thank you very much for this very interesting talk. So my question is, would you consider at some point trying to advocate including HCA as one of the sustainable developmental goals? Because I mean, underdeveloped countries are very familiar with funding instruments that are derived from such programs. They already have infrastructure and things like that in place. Maybe that could be a way to encourage them to participate?
Yeah, my brief answer to that is that I think this is the push to disease in terms of using HCA and building HCA disease data sets as part of the SDG goals in terms of human diseases. So I think that's a very well taken point, and that's a vehicle for us to do that. Chloe, you wanted to say something? Sorry, can you make that mic live? That one, pink mic.
Pink mic. Okay. I was hoping Partha would add on this prescriptive list how we can better partner with different commercial entities to actually really not only help your infrastructure but also access to reagents. You know, in interacting with all of you, I have a whole new appreciation of how it takes a really long time for you to not only get your reagents, but actually, because of the challenges of distributors, sometimes it ends up costing you even more. So I was hoping someone would mention this because you have a unique opportunity in this meeting to start, you know, interacting with more commercial partners, and we can, you know, try to kind of come together to try to find better solutions.
Thank you for saying that, Chloe. I do know that there are a number of networks and people in this room from South America, some Latin America, and also from other regions of the world who have actually engaged directly with Illumina and other suppliers to try to really reduce the cost of these sorts of things and reagents. I just want to give Shyam, who's the chair of this diversity task force, the last word before we close this really interesting and important session.
Thanks, Musa. Quickly, 2 points. One is there'll be 4 talks after this. Look forward to that, people giving their experience. There will be panel discussion after that, so hopefully to get your feedback and ideas after that as well. And on a positive note, I hope you're convinced diversity is a scientific frontier. It's untapped. There's huge amount of exciting work to do. Diversity influences our biology more than we could imagine. And so we're going to have a lot of fun working on this.
Thank you, Kian. Thank you for attending this session. Thank you.